Okay, thank you. Yeah, so I'm going to talk about quantifying larval fish abundance using DNA metabarcoding. So because this is generally a conservation-focused session, I'm going to first want to just talk about why it's important to know what's going on at early life stages for fishes. One reason that's really useful for this is detecting where invasive species might have been established or more recently established from novel detections of larval fishes and eggs showing they're reproducing there. But it's also really useful for identifying important habitat for sensitive species or species at risk for their nursery habitats, as well as determining where along this life cycle human impacts are affecting the abundance of fishes that are really important for fisheries. So whether human impacts are affecting them at their adult stage or at juveniles or larval stages. However, to do this correctly, you need to do a whole lot of sampling. So you have to catch lots of larval fish over a large period of time because each species can go through those life cycles at different times of the year. They can also do that at different times in the habitat in different places. So what you get left up with is a whole lot of larval fishes that all look almost the exact same. So identifying the differences between those fish is really difficult. Now DNA barcoding has made that quite a bit easier, but you're still often limited by how many individuals you can process if you're using just standard SING or sequencing. Now, DNA metabarcoding makes that easier as you can process many individuals at the same time. So you can take a sample containing those 4 fish instead of processing them independently, blend them all together, and sequence that homogenized sample. This allows you to process way more species, which should give you more overall accurate estimates of your abundance of larval fishes by expanding your surveys. However, there's a lot of issues with DNA metabarcoding that could really impact these estimates, and this often comes down to the possibility of species-specific biases. So this will happen whether or not you're looking at larval fish from homogenized samples or from taking eDNA samples from a region where you expect to find larval fishes. And there's other issues that you have to consider if you're taking eDNA samples, like differentiating larval fishes from adults. But I'm not going to get into that today. I'm going to talk more about the DNA metabarcoding aspects and properties of that is pretty general across all studies that need to be incorporated and understood if you're going to understand how those biases can impact abundance estimates. So for larval fishes, if you're blending them up, larger fish would contain more DNA and as a result would be overrepresented in your sample based on if you're looking for numerical abundance. As we know from eDNA from some of the other talks, depending on the abundance of those individuals and the shedding rates and surface area of those individuals, you might also have a biased output of DNA into the environment. And as well, primer bias can have a pretty significant impact on this, with primers preferentially binding and amplifying certain sequences more readily than other ones in the sample. So when you look at your read abundance at the end of the sequencing analyses, you end up getting some species overrepresented than others. So I think what's important to know before we start applying this methodology to these surveys is whether the advantages of taking more individuals and sampling for more individuals in the environment actually or isn't totally jeopardized by some of these other species-specific biases that could really impact your overall estimates of abundance for these fishes. So my 2 main questions were how accurate is DNA metabarcoding for estimating larval fish abundance for bulk samples of DNA? And does DNA metabarcoding improve overall accuracy of these studies? And before I get into the specific samples I'm going to look at today, I want to talk a little bit about the habitat that we were sampling for this. So these fishes were collected in 2021 by the Saugeen Ojibwe Nation's Coastal Waters Monitoring Program. And it was from this part of the Saugeen Peninsula near Stokes Bay where these studies have been done over a long period of time. And that's because this is important nursery habitat for ciscos and lake whitefish, which are really important culturally, economically, and nutritionally for the Saugeen Ojibwe Nation. So they really want to know what's going on at the larval stages for those fishes. But to get the most out of each one of those samples that we collected, and I'm not going to talk about all the other interesting things that we're doing with those samples, but to get the most out of them, we took a subsample of them, so 33 of these samples, to investigate more how we can incorporate this DNA metabarcoding analysis into future studies. As well as better understand how that methodology and the biases in that methodology will impact these results. So one of the interesting things about these samples, well, there's a few interesting things that I want to point out early on. So the one thing early on in the season, you get the co-occurrence of a lot of ciscos and lake whitefish in the same pulls of the larval fish bongo nets that they pulled along that bay there. What I want to point out is that you typically see lake whitefish being larger than the ciscos in those same samples. So the red dots higher up on that y-axis there, which is total length, than the ciscos at the same time. I also want to point out that ciscos are generally more abundant, so the majority of those dots are blue. And as you go over time, the larval fish get a little bit longer, as you might expect, and also start to co-occur with other species like yellow perch and Iowa darter. The last thing I want to point out is that for some of those samples, you can pull these nets through the water and you end up with a whole bunch of individuals, but they're all the exact same species. And that just happens to be of the nature of ichthyoplankton floating around in the water at this time and at that place. So that's not uncommon for these kind of larval fish nets, and just something that's going to come up again later. So the first question I wanted to talk about is how accurate DNA metabarcoding is. And to test this, I took this approach. So the results that I showed you just on that page previous were done by taking one half of the fish and processing that separately to get the true numerical abundance of each species in that sample. So those are all sequenced separately, and you would get the abundance estimates from that. And then took the other half of the fish, homogenized that together, and took a bulk sample of that DNA homogenate, and used a two-step PCR approach with the MyFishU primers to get DNA metabarcoding results. I then took the relative proportions based on those read frequencies I got for those different species in that sample and compared that back to the true abundance to see how off those estimates were from the expected numerical abundance of those fishes in those samples. At last, after that, and my little dots aren't showing up, but I took the ethanol that those fishes were stored in, filtered that, and processed that like I would process an eDNA sample. So this is essentially a very small, I guess, eDNA sample of fishes that were sitting in that jar. I did the same thing that I did with the bulk tissue sample, used the MyFishU primers, processed that, and then compared those reads back to the true numerical abundance for that sample. And again, got an estimate of how far off that was from the expected proportions. So the results from that are right here. So I'm just going to step through this from left to right. So on the very left, I have the homogenized tissue, and what you'll see there is that the lake whitefish were often overestimated based on the number of sequencing reads when you compare that to the proportion that you would expect based on the true numerical abundance in that sample. And that makes a lot of sense, given that those fish were almost always larger than the ciscos that were collected in those same samples. I want to point out that there's that one blue dot very far to the left-hand side for the cisco. That was found in the same sample as the yellow perch, where they were almost 3 times as long as the yellow perch in that sample. So it makes sense that it was very much overestimated based on the amount of DNA that those larger larval fish would have. Now this trend breaks apart when you start looking at the ethanol samples, so the kind of proxy I have for eDNA. And not only does the estimate or the median estimate of that get closer to 0, so no difference between the expected and the observed reads, but it actually switches in the trend, with the ciscos being overestimated relative to the lake whitefish. And if you remember from the previous one, ciscos were often more abundant. There was often lots more ciscos in a sample than lake whitefish, despite the fact that they're smaller. If there's lots of small individuals, they'd have more surface area. So that's possibly the explanation for that. I don't really see any evidence of primer bias. You wouldn't really expect a lot of evidence for primer bias from lake whitefish and ciscos based on how similar they are in their sequence. I think there's 1 or 2 base pairs difference in the amplicon that is generated for them from the 12S primers, and there's no variation in the primer binding regions. So obviously you don't want to see any bias when you're doing these kind of analyses, because if you're trying to predict an overall abundance from these smaller subsets of samples, that could really bias your overall estimates. But there is some interesting aspects. You could kind of take these approaches in tandem to try to get both an idea of numerical abundance and biomass-based abundance based on this. But I think it's also important to consider the system that you're looking at and how these biases might affect that. So if you remember from the plot, one of the other aspects of these surveys is that you could get these samples where you have a large number of individuals, but all of one species. And these are essentially freebies, because there's no bias in those bulk samples because it's all just one species. So if you're looking at an overall survey of the number of larval fishes that you're collecting, those samples work really well. And you would get accurate counts of the number of individuals of each species in those. So I think it's important to kind of consider that when you're looking at this from a practical perspective of people who want to know how many larval fish are in the habitat that they're surveying. Now it breaks apart a little bit when you have more than one species and that bias is present, but because so many samples contain just one species in this habitat, it works out pretty well. So I wanted to look at this a little bit more in detail. So I took a simulation-based approach to test this. So what I did is I took some previously published surveys for larval fishes containing lots of data from different surveys, including the one that I just talked about, and I subsampled individuals from the results of those surveys, so the count data from those larval fishes, subsampled 1 to 100 individuals, or took an equal number of extractions or number of samples of the specific sampling points, and I processed those in a simulation of a PCR. So that simulation of a PCR included all of those biases that I previously found from the from the experimental studies I did, as well as some biases associated with subsampling DNA from a larger sample and differences in the template that you would have. I then took what estimate I would have if I increased that to what I would expect for the total number of larval fishes collected in those samples to see how far off it was from taking random individuals from across the survey or taking an equal number of samples or an equal number of extractions of the bulk samples, assuming some bias in those estimates. Now I'm just going to skip through this because I think I'm running out of time, but I will mention that those surveys were super different in both the number of samples that were taken, the number of species in each one of those samples, and the number of individuals in each one of those samples. Now for the Stokes-Bayes samples that I just talked about, there wasn't a whole lot of difference if you took random individuals relative if you took a by sample approach. The sample approach with some bias still worked a little bit better, but not significantly so. And same is true for the Bruce Power sample. So these are from entrained larval fishes at Bruce Power. And in the same case, there wasn't a whole lot of different species, not a ton of individuals per sample. And you also don't see a ton of change. But if you look at a really large survey where there's tons of individuals per sample, lots of different species per sample, you start to see a better or a better result when you start doing a by sample approach, even though there is some bias in that, than you do by taking just a small subsample of individuals, probably because there's so many individuals to look at. Does it still work? Okay. So homogenized tissue, I've shown as homogenized tissue reflects the biomass-based abundance more so than the ethanol-based approach, which looks more at, seems to produce more numerical abundance results. So I think this is important when we start to think about doing eDNA for this kind of study. And despite the biases that you see, it seems that it still doesn't jeopardize all the advantage that you get from being able to process way more individuals, which is known to be really important for larval fish. So with that, I'd just like to thank some of the work studies who were involved in this, the Coastal Waters Monitoring Program for the sample collection, and some funding from MyTax, GenFish, and the Nuclear Innovation Institute. Thank you.